Bismillahirrahmanirrahim. I am Dr. Abdul Wafar, and the topic of discussion is transcription. What is transcription? Transcription is copying process of DNA molecule in the form of working copies. It is just like photocopying DNA molecule at a specific page. Just like we photocopy a particular page of a book, transcription is copying the DNA molecule at a specific point for a specific gene. Transcription synthesizes RNA molecule using DNA strand as a template so that the genetic information can be transferred from DNA to RNA. So RNA is working copy. This is transcript is a message that comes from nucleic acid to ribosomes to synthesize a particular protein. Similarity between replication and transcription. Both processes use DNA as a template. Phosphodiester bond formation takes place in both of the processes. Both synthesis directions are from 5' to 3'. There are certain differences in replication and transcription. Replication template, both the template strands, they are copied in replication, while in transcription template is only one strand. One strand is copied at one time. Substrate is DNTPs, deoxynucleotide triphosphate, while in transcription it is nucleotide triphosphates. Means it is ribonucleotide triphosphate, deoxyribonucleotide triphosphates, and ribonucleotide triphosphates. In replication, we use primers. In vivo, primers are required, while no primer is required in transcription. DNA-dependent polymerase used in replication, while DNA-dependent RNA polymerase is required in transcription. Product is double-stranded DNA in replication, while single-stranded RNA is product in transcription. Replication in base pairing takes place adenine and thymine, cytosine and guanine, while in transcription, adenine and uracil, thymine and adenine, and cytosine and guanine. So there are some differences where nucleotides are uracil in RNA, while thymine in DNA. So where adenine is present, thymine will not be added, uracil will be added for transcription. Where thymine is present in DNA, adenine will be added in front of thymine, guanine and cytosine. Templates and enzymes used in transcription. The whole genome of the DNA needs to be replicated in replication, but only small proportion of the genome is transcribed in response to the development of the required physiological response or required product. Which product is required? Its gene is activated for transcription. Only one specific gene is transcribed. So there are multiple genes which are transcribed many times. This is a continuous process in the cell which takes place every time within the cell. But it doesn't require copying of the whole genome in the form of RNA. Only a specific part is copied. DNA regions that can be transcribed into RNA are called as structural genes. Means only structural genes, they are copied in the form of transcript, while rest of the chromosome or genome is not copied. What is a template? Template strand is the strand of the DNA from which RNA is actually synthesized, is transcribed. It is similar as template present in replication. It may be also named as antisense strand. So I will later discuss sense and antisense. Template strand is copied in the form of messenger RNA, and the coding strand is the strand whose base sequence specifies the amino acid sequence of the encoded protein. Coding strand is actually complementary strand of the template strand. It is similar to RNA synthesized, and it is also called as sense strand, means the strand of the nucleic acid which acts as template. Its opposite strand is called as coding strand. Why we call it as coding strand? Why we don't call template strand as a coding strand? It is because the messenger RNA synthesized, it has codons that specify certain amino acids. These codons, they have same sequences as complementary strand to the template in DNA. So the complementary strand of DNA and messenger RNA, both of them have same sequence except difference between thymine and uracil and deoxyribose and ribose. So messenger RNA has same sequence as complementary strand of the template strand. That's why the codons present in messenger RNA, they have same sequences as compared to the complementary strand of the template. So the complementary strand is called as coding strand. It is also called as minus strand. It is also called as sense strand and non-template strand. While template strand, positive strand, anti-sense strand, and non-coding strand, these are the names of uh, template strand from which messenger RNA is produced as a complementary copy. So look at this. This is a coding strand and template strand. Both of them have same sequences as compared to only the difference is adenine and thymine. So this is messenger RNA. It is synthesized from another strand which is present below this strand, that will be template strand. While that template strand, if represented in black, it is complementary to this one. So this is complementary strand, below is template strand. Sorry, this is coding strand and this is template strand. Red is template strand and this is coding strand. So if we compare the sequence of this messenger RNA, this is produced from this template strand. This is template strand. So its complementary sequence is present, except adenine with uracil, guanine with cytosine. If we compare this blue sequence with this black sequence, that is non-template strand, both of the sequences are similar except the difference of thymine. So if we think this is a code, GCA is a code of an amino acid in messenger RNA. So GCA is also present in non-template strand. This is non-template strand, this is template strand. Messenger RNA is synthesized from red and this is black is non-template. So this has similar sequence. So this is a code. That is why this strand of the DNA, which is complementary to template strand, we call it as coding strand. In bioinformatics, you will find the sequences of DNA given in the form of coding strand, non-template strand. Always sequence present in the bioinformatics is in the form of non-coding strand, non-template strand. So this helps the prediction of messenger RNA. Just replace this strand with uracil, thymine replaced with uracil, you will get sequence of messenger RNA. What will be messenger RNA produced from coding strand? Just replace uh, thymine with uracil. You will get transcript, primary transcript. You will get messenger RNA. So this is coding strand. This is the below purple is template strand, template strand, template strand, coding strand, coding strand. For example, if this is a template strand, this is its coding strand. If we call this as a template strand, this will be coding strand. Why one time it is called as template, the other time it is called as coding? Because genes in chromosome, they are present on both the strands. So one strand may act as coding strand at one time, and its one strand may act as template strand at one time, and its complementary strand will be called as coding strand. While at another time, when we want to copy the gene present in other strand, when it is being copied, it will be called as template strand, and its complementary strand will be called as coding strand. So genes are present in both directions, in on both strands of the nucleic acid of the chromosome. So coding information is present in organization of the coding information in viral genome. In one direction there may be certain genes, in other direction there may be certain genes. What is RNA polymerase? RNA enzyme, the enzyme responsible for the synthesis of RNA is DNA dependent RNA polymerase. In prokaryotes, RNA polymerase is a multi-subunit protein of 480 kilodalton. It means it is a very large protein. Eukaryotic systems have three kinds of RNA polymerases, each of which is multi-subunit protein and responsible for transcription of different RNAs. When this enzyme is complete catalytically active, RNA polymerase, DNA-dependent RNA polymerase, when it is complete catalytically active, it will be called as holoenzyme. Holo for whole enzyme. Complete catalytically active enzyme is called as holoenzyme. It consists of 5 different subunits, 2 alpha, 1 beta, and 1 beta prime, omega, and rho. So look at this enzyme. 2 alpha, 1 beta, another beta prime, and there is omega on in blue on backside. Rho, this is core enzyme. This is an enzyme which is not complete catalytically active. So there are 1, 2, 3, 4 different type of subunits. When fifth subunit, that is rho factor, binds to this enzyme, it becomes complete catalytically active. Then we call it as holoenzyme. So this is holoenzyme. Look at the difference. This is core enzyme, this is holoenzyme. Holoenzyme is complete catalytically active enzyme. Subunits. So there are subunits represented. So there are some inhibitors of drugs which inhibit this synthesis of RNA in bacteria, like rifampicin is an anti-tuberculosis drug. We use it to treat tuberculosis, Mycobacterium tuberculosis. So if we use this drug, it inhibits synthesis of RNA in prokaryotes. RNA polymerase of the other prokaryotic system is similar to that of E. coli in structure and function. So RNA polymerase of eukaryotes. Eukaryote polymerases, there are 3 different polymerases in eukaryotes, polymerase 1, 2, and 3. So that transcribes these different types of RNAs. We have different RNAs in eukaryotes. There are 4 different types of messenger RNAs. There are ribosome. There are 4 different types of ribosomal RNA, 5, 5.8, 18, or 28. While in prokaryotes it is 3 types. Similarly, transfer RNAs, 40 to 50 different types of transfer RNAs, and messenger RNA. There are small nuclear RNAs. There are so different enzymes are present in prokaryotes. One alpha amanitin present in fungi, it inhibits RNA polymerase. RNA polymerase 3 and 2. So amanitin is a strong inhibitor and leads to death if this uh, mushroom is eaten mistakenly by someone. It immediately inhibits transcription throughout the body, and that leads to death. So some mushrooms, they are toxic and they are dangerous for health, for life-threatening mushrooms. Replication takes place at a particular point. We call it as recognition of the origin of transcription. Sorry, transcription also takes place at a specific point. Similarly, transcription has three steps: initiation, elongation, and termination. So initiation takes place by recognition of origin of replication in the chromosome or genome or nucleic acid. Replication also takes place in prokaryotes as well as in viral RNA. When it is embedded into host genome, it replicates, it transcribes. Uh, so there is a particular region, we call it as operon, open reading frame, several structural genes and regulatory sequences. Uh, One operon, that is lac operon in bacteria, it is it it consists of three genes. Uh, Before these genes, there is promoter. That promoter in prokaryotes, it controls expression of these genes. So promoters are actually the regions which are recognized by uh, row factor of uh, RNA polymerase. So there are conserved regions of polymerase. We will discuss it. What are promoters? There are conserved regions of promoters. Promoters, prokaryotic promoters, they are present before structural genes. These are this is a structural gene. There may be one structural gene. There may be cluster of genes present downstream to a particular promoter, and these are regulatory sequences. These regulatory sequences, they are promoters, and promoter is recognized by RNA polymerase, the last factor, sigma factor. Prokaryotic promoters, they have certain particular characteristics. Likewise, eukaryotic promoters, if this is start of the transcription, on downstream, this is downstream. On downstream, it is plus sequence, and on upstream, it is minus sequence. All the nucleotides present before the start of the transcription, they are represented with minus, like minus 1, 2, 3, 4, 5, and 10. And on right side downstream, it is represented with plus sign. So there is a minus 10 region present in prokaryotic promoters. We call it as Tata box. Tata, T-A-T-A, Tata box. So this sequence is present 10 nucleotides, approximately 10 nucleotides before the start of the transcription. There is no zero nucleotide. Minus 1, plus 1. This Tata box is also called as Pribno box. There is minus 35 sequence present in prokaryotic promoters, TTGACA, minus 35. These two conserved regions, they are present approximately in all promoters of the prokaryotes. These are called as consensus sequences. Look at these regions in prokaryotes. So this is these are different genes aligned on to one another. So there is a spacer of approximately 5 to 8 or 7, 10 or 9 nucleotides. There is minus 10 region, and that region is TATAAT in prokaryotes. There is a spacer of approximately 17 to 18 nucleotides, and then if we add all these nucleotides, it becomes approximately 35. So there is minus 35 sequence. So there are some variations. However, most of the sequences, they are conserved. These two regions, they are conserved. Minus 35 region of sequence is recognition site of the binding site for RNA polymerase. Minus 10 of the Tata box is the region which is a stable complex of the DNA and RNA polymerase is formed. How transcription takes place? There are general concepts, 3 phases: initiation, elongation, and termination. The prokaryotic polymerase, RNA polymerase, can bind to DNA template directly in transcription process. Eukaryotic polymerase requires cofactors to bind. So there are initiation factors in eukaryotes which bind to this region and they synthesize. So there are initiation factor 1, initiation factor 2, and 3 in eukaryotes, while in prokaryotes, it doesn't require an initiation factor. But prokaryotic polymerases never requires a primer as well as eukaryotic DNA-dependent RNA polymerase. So all DNA-dependent RNA polymerases, they don't require any primer molecule. Primer is always RNA in nature or sometimes DNA in vitro. But in transcription in vivo, never required primer. Transcription of prokaryote, initiation phase, there is elongation phase, and there is termination phase. Initiation phase, RNA polymerase recognizes the promoter and starts the transcription. So in initiation, RNA polymerase recognizes one segment, recognizes, binds to promoter region, opens it, and then transcription starts. Then elongation, it is the RNA strand is continuously growing. And in termination, RNA polymerase stops synthesis and nascent RNA molecule, newly synthesized RNA molecules separated from DNA template. Initiation, RNA polymerase recognizes the -35 sequence TTGACA region and slides to the Tata box region. Then it opens the DNA duplex. Means it binds to this region and then it slides to Tata box. While sliding, it opens the double helical strand and then double helical DNA is opened. It unwounds approximately 17 base pairs. The region is unwound. So look at this. RNA polymerase sigma factor binds at this region, slides to this. So this is a region -35 TTGACA, and then it slides to Tata box. While moving in this direction, just it is embedded inside, like it opens the double helix, just like zip opens, zip is opened. The first nucleotide of RNA transcript is always purine triphosphate. GTP is more often than ATP. GTP is more often than ATP. The structure of RNA transcript, it continues. 3 molecules form a transcription initiation complex. No primer is needed. Sigma unit binds and falls off from the RNA polymerase once the primary phosphodiester bond is formed. Means sigma factor first binds, recognizes, opens it, and calls for rest of the 4 subunits to complete catalytically to start the replication transcription. So when first 2 nucleotides, they get bound, this subunit is separated out and the core enzyme moves along with the template in elongation phase. Elongation, the release of the sigma subunit causes the conformational change of the core enzyme. The core enzyme slides on the DNA template towards 3' end. Likewise, RNA is also synthesized from 5' to 3'. So new RNA is synthesized from 5 to 3, while template it is synthesized from 3 to 5. So this is addition of nucleotides. So this is n number of nucleotides, NNTP nucleotide triphosphate like adenine. It joins this and N+1 PPI. RNA polymerase DNA segments of the approximately 40 nucleotides and then nascent RNA from the complex is called as transcription bubble. So a bubble is produced. I will show you later. 3' segment of the nascent RNA hybridizes with the template toward the 5' end and transcription bubble synthesized in the process. So this is actually a transcription bubble. So first it was bound at this region. This is a transcription bubble. This is non-template strand, this is template strand, this is also mm, coding strand, minus strand, non-template strand, and this is template strand, this is non-coding plus strand. So this unwinding takes place in this direction. Similarly, gyres or topoisomerases, they unwind this uh, parent DNA molecule template, and there is another gyrase which rewinds this. So while rewinding, this RNA molecule, newly synthesized RNA molecule, comes out of this transcription bubble, and this whole complex moves in this direction. While this is template, this is green is newly synthesized messenger RNA. It comes out of uh, this transcription bubble. This is RNA polymerase of E. coli, different subunit. This is purple is DNA molecule. Green is RNA molecule. So this is how it binds uh, to transcription start site promoters. This is prokaryotic promoter, +1, -1, -10, -35. So this is sigma factor that binds to this region. Closed complex, when it exerts some and it slides in this direction, it opens this double helical. So this is initiation, open the complex, transcription, initiation. So this is transcription, initiation. I will go to back slide. Look at this region. There are two, it looks like two molecules, but this is sigma factor. First it binds to this, then this whole complex binds. So first it binds this region, then these are the four other molecules, 2 alpha, 1 beta, 1 beta prime, and omega. It binds to this region. So once transcription starts, this sigma factor, it separates out. So this factor is actually different for all the genes. So how to get information, which gene is to be transcribed, this is decided by this sigma factor. This sigma factor gets some information within the cell from certain signals, like if glucose molecule is high, it gets some information from insulin, it becomes activated, it is phosphorylated, and it binds uh, to the promoters of the genes involved in glucose metabolism, like phosphofructokinase molecule enzyme is synthesized as a result of this transcription. Termination. RNA polymerase stops moving on the template when it reaches certain destination. Termination of the transcription falls off from the transcription complex. The whole complex moves off from the transcription complex. So there are two types of termination. One is row dependent termination, the other one is row independent termination. Termination, the termination function of row factor. What does row factor? Row factor is a protein that binds to newly synthesized messenger RNA and stops the polymerase, gives some message to RNA polymerase that to stop the transcription. ATPase and helicase. Hexamer is a ATPase and helicase. There is row independent termination. How it works? Row independent, the termination signal is stretch of 30 to 40 nucleotides on RNA transcript. So this signal comes from template strand. You know, RNA synthesized from templates. So there are certain sequences which are present on DNA at the end of the gene that is rich in thymine and adenine. So that incorporates series of transcript nucleotides on the RNA transcript consisting of many GC. So if adenine is present, cytosine in thymine in uracil in messenger RNA. So it is followed by a series of uracil in messenger RNA. The sequence of the specificity of this nascent RNA transcript is from particular. So it makes a loop structure for the termination transcript. Loop, like hairpin-like loop, is produced in row independent termination. Look at this. This is RNA. This is DNA. This is newly synthesized RNA. This RNA makes a loop like hairpin-like structure. So this pin acts like a stop gear, brake, handbrake. So it produces brake-like structure in the messenger RNA, any RNA, messenger RNA, any other RNA. So this gives message to RNA polymerase to stop transcription, and then whole machinery dislodges from the complex, transcription complex, and RNA comes out of the. So this is a hairpin-like structure produced in messenger RNA itself that halts the activity of the RNA polymerase so that it stops the transcription process, and that leads to termination of this RNA transcription. Stem loop stem loop structure alters the conformational change of the polymerase. So when it is bound at this region, it makes conformational change in the RNA polymerase. So DNA RNA hybrid stability decreases, and that causes dislodging of the nucleotides. Transcription in eukaryotes. So there is initiation, there is elongation and termination. In similar way, cis acting elements, they act as in initiation of the transcription, and there are cis acting elements. Again, we are aware that eukaryotic genes, they are composed of introns and exons. So structural gene in exons are the region of the gene which specify certain amino acid, while introns, they don't specify certain amino acids. So this is start of a gene. There may be many introns in a gene. There may be single intron or no intron in the gene, like histone proteins. Genes, they don't have any intron, while 11 introns, 10 introns are present, 11 exons are present in gene of albumin in eukaryotes. So these are the structural genes, and there are regulatory genes. So there is a Tata box. There is Hogness sequence. There is a cat box. This is approximately 25 nucleotides. Before the start, there is a GC box, approximately 80 nucleotides. There are enhancer elements. So look at the complexity of the eukaryotic gene. There is a start site. There is Tata box, approximately -25, cat box -40, GC box -80, but it differs in species to species. So this Tata box is called as Hogness sequence. This is cat box, CAAT, you mean big cat, CAAT cat. And there is GC box. So all these are called as cis acting elements. Look at Tata box. So this is -10 and so on. Okay, so there are different genes. Transcription factors. Transcription factors, they allow initial binding of the DNA polymerase 2 with the template DNA. So these are transcription factor 1, transcription factor 2, 3, like transcription factor 2A, transcription factor 2H. The trans-acting factors are the proteins that recognize and bind directly or indirectly cis-acting elements and regulate its activity. So there are certain transcription factors, transcription initiation factor, a lot of initiation factors present in prokaryotes. So they catalyze RNA synthesis, specify recognition of Tata box, stabilize DNA binding, binds to and recruits polymerase 3 to binds tightly And elongation factors, there are certain elongation factors. Likewise, there are certain termination factors as well. Pre-initiation complex. So the initiation complex on Tata box binds to, first of all, this protein binds, then rest all the proteins bind, helicase activity, H. Pre-initiation complex. So this is Tata box, TBF and TAF bind to this region. Transcription factor 2A helps this binding, then transcription factor 2B, and then polymerase 2, transcription factor 2F. So look at the complexity of initiation in eukaryotes for transcription. So this is polymerase 2 in eukaryotes. It binds to this region. Transcription factor 2E binds to this region, H, and phosphorylation of polymerase, RNA polymerase. When phosphorylated, these molecules become active. So C-terminal domain of the CTD is C-terminal domain of the RNA polymerase, and it moves towards the downstream, starting the elongation phase. Transcription factors fall off from the pre-initiation complex during the elongation phase. Elongation is similar to that of the prokaryotes. The transcription and translation don't take place simultaneously since they are separated by nuclear membranes. While in prokaryotes, it is simultaneous, because in prokaryotes, life of the cell is 20 minutes. So they have to replicate simultaneously, they have to transcribe, synthesize certain proteins. While in eukaryotes, transcription takes place in nucleus, while translation takes place in cytoplasm, cytoplasm where ribosomes are present. So simultaneous, these processes, they don't take place simultaneously. Newly synthesized RNA comes out from nuclear pore, and it comes out to cytosol, where it is translated. So look at the polymerase and how it is moving in forward direction and synthesizes RNA. Termination is AATAA. So look at the repeated poly A tail at messenger RNA. Post poly A tail followed by GT repeats. Termination sequence in so this sequence is present in DNA. AAT. So I already told that certain sequences rich in adenine and thymine. Adenine and thymine present in template DNA. So this will lead to poly U addition at end of the messenger RNA, followed by GC repeats. Termination is closely related to post-transcriptional modification. So this is all about transcription, and then when transcription completes, it leads to the synthesis of mature messenger RNA. Primary transcript is modified in post-transcriptional modification. So there are many post-transcriptional modifications in eukaryotes. Nascent RNA known as primary transcript needs to be modified to become a functional molecule like transfer RNA, ribosomal RNA, messenger RNA. The modification is critical in eukaryotic system. So there are certain transcription modifications: capping at 5' end, tailing at the 3' end, messenger RNA splicing to remove the introns, self-splicing. So this is a mechanism which recognize that RNA molecules, they also act as enzymes. So we call them ribozymes. So this is the first clue that represented that RNA molecule was first present, then it was transcribed and translated into proteins. So that is, life started in the form of RNA initially. So this is how, whether egg was first or hen was the first to come on the earth. So there is RNA editing in three-dimensional structure, and removal of the RNA 5' capping, and editing in the form of to coil it into three-dimensional structure for to be bound at ribosomes. So this is all about translation. I will not go into much detail of this capping. Actually, this capping is in opposite direction. Look at both the 5' ends are joined to one another. So 5' capping, it is called as 5'. It is, this nucleotide is joined in opposite direction. It should have this 3' end joined at this region. All the nucleotides are joined to 3', 3', 5', while only this 5' capping in messenger RNA eukaryotes is bound in opposite direction. So this is very resistant to RNA polymerases in the cytosol. So this protects the cell, protects the newly synthesized messenger RNA to be degraded by RNases in ribosome. So this is 7-methyl guanosine cap present on cap of messenger RNA on 5' end. 7-methyl group is attached, 7-methyl guanosine. So there is poly-A-tail, addition of polyadenylation. This polyadenylation by poly-A polymerase enzyme. So that adds so many adenines residues at 3' end of the newly synthesized messenger RNA to make poly-A-tails. This decides for how many times this messenger RNA will be translated. So there is messenger RNA splicing. So introns, they are removed through a self-splicing mechanism in which there is -19 intervening sequence. There are, so these introns, primary transcript is copied as such from template DNA. So if template has so many introns or even one intron, that should be replaced to make a mature messenger RNA for translation. So this is synthesized in the form of, this process is carried out by self-splicing of the RNA molecule. Thomas Ketch was the first scientist who discovered this phenomena of self-splicing to discover ribosome in tetrahymena thermophila. So this is a split gene. So this gene is coding and non-coding regions. A, B, C, D, E, and F, and G. These are non-coding and coding regions. A to G non-coding regions and A to G non-coding regions, while this is coding region. 1, 2, 3, 7 regions. So approximately 7,700 base pairs, but much of it is non-coding region, exon and introns. So introns are removed through splicing mechanism, RNA splicing. So this is all about the lecture. That's